Welcome back, students. Today we'll be revising on the topic structure of nucleic acids. Now, in this module, I'll be telling you about the basic structure of nucleic acids, structure of DNA, and the packing of DNA in both prokaryotes and eukaryotes. Now, as you know, there are two types of nucleic acids. One is the DNA, that is deoxyribose nucleic acid, and the other is ribose nucleic acid. Now, these nucleic acids are made up of basically three components, namely uh, sugar. It is a pentose sugar having formula C5H10O5 in case of ribose sugar, and in case of deoxyribose sugar it is C5H10O4. Now, the sugar that is present in RNA is ribose, and the sugar that is present in DNA is deoxyribose. Now, at the carbon 1 position of this sugar, a nitrogen base is attached, which is the second component of a nucleic acid. Now, these nitrogen bases are of four types. That is, in DNA, we have adenine and guanine, thymine and cytosine. Adenine and guanine are purines, and thymine and cytosines are pyrimidines. The nitrogen bases of RNA are adenine and guanine, that is, the purines are same both in DNA and RNA, but one pyrimidine is different. That is, in DNA we have thymine. Here, in case of RNA, in place of thymine we have uracil, and the fourth nitrogen base, that is a pyrimidine cytosine. Now, when a nitrogen base gets attached to a sugar molecule, you get a formation, that is a nucleoside. Now, to this nucleoside, if the third component of the nucleic acid, that is the phosphoric acid, gets attached, then it gives rise to a nucleotide. Actually, nucleic acids are a long chain of these nucleotides. Now, one nucleotide and the other nucleotide, the successive nucleotides, they remain attached by a phosphodiester bond established between the fifth position of one nucleotide and the third carbon position of the successive nucleotide. So always there is a phosphodiester bond. One end is the 5 end, the other end is the 3 end. So this gives orientation to the nucleotide chain. So these nucleic acids are nothing but a long chain of such nucleotides, where the phosphate and sugar, they form the backbone, and to this the nitrogen bases are attached at right angles to the sugar molecule. Now we shall see the structure of DNA. DNA consists of two molecules that are arranged in a ladder-like structure called a double helix, because DNA always is a double helical structure. A molecule of DNA is made up of millions of such tiny subunits called nucleotides, as I've told you earlier. Each nucleotide is made up of a pentose sugar, a nitrogen base and a phosphate group. Now this is the structure of a pentose sugar of DNA. Now here you will see at this carbon 2 position there is one oxygen atom less. That is why the name is deoxyribose sugar. And it is because of this absence of one oxygen atom, it gives more stability to DNA molecule. It becomes less reactive. The four nitrogen bases of DNA. Now these are the pyrimidines. Pyrimidines are single ring structures. Now this is a structure of a cytosine molecule. This is a structure of a thymine molecule. And these are purines. Purines are double ring structure. That is, to this benzene ring like structure, there is a side imidazole ring present here. Now always the third carbon position of a pyrimidine gets linked to the first carbon atom of sugar, and the ninth position of a purine gets attached to the first carbon of a sugar molecule. That is how the nitrogen bases get attached to the sugar molecule to form a nucleoside. Now, when a pentose sugar and the nitrogen base get attached, as I've told you, you get formation of a nucleosides. The four nucleosides of DNA are deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine. Now to this nucleoside, DNA nucleoside, if a phosphate molecule is attached at the fifth carbon position, you get formation of a nucleotide. The four nucleotides of DNA are deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. Now these can also be called as deoxyadenalic acid, because the acidic property to the nucleic acid is because of this phosphate group. So if this phosphoric acid is attached to the deoxyadenosine molecule, you get deoxyadenalic acid. Similarly, we have this deoxyguanilic acid, deoxycytidalic acid, and deoxythymidalic acid. Now, these four nucleotides, they show complementary pairing. And because of this complementary pairing only, the two strands of DNA are attached with each other. Now, adenine always pairs with the thymine, and cytosine always pairs with the guanosine. This pairing was actually proved by the work done by Chargaff. When Chargaff was studying the amount of these nitrogen bases in different molecules, he came into this conclusion, and these conclusions we refer them as Chargaff's rule of base equivalence. According to this, the amount of purines is always equal to the amount of pyrimidines. That is, amount of adenine and guanine in any organism is always equal to the amount of thymine and cytosine. And second, the amount of adenine is always equals to amount of thymine, and amount of guanine is always equals to amount of cytosine. It is because of this result he could suggest that adenine always pairs with thymine and guanine always pairs with the cytosine. And this ratio, that is the amount of adenine and thymine to the amount of guanine and cytosine, is always constant for a particular species. And the deoxyribose sugar and phosphate, they occur in equimolar proportions. Now, because of this complementary base pairing, the order of bases in one strand determine the order of bases in the other strand. Now here there is one strand of a DNA molecule, and this is another strand of DNA molecule. Because of the complementary pairing, we can easily say amount of adenine is always equal to amount of thymine. Now here you can see adenine molecules are 1, 2, 3, 4. Similarly, thymine are also equal. Now, the two chains of DNA are always running anti-parallel. Now, whenever this pairing occurs between this adenine and guanine always pairs with cytosine with 3 hydrogen bonds, and adenine always pairs with thymine with the 2 hydrogen bonds. Now, one strand you see the phosphodiester bonds are in the direction 5:3 in left to right, and in the opposite strand you can see the phosphodiester bond direction, that is from 5:3, is from right to left. So this shows that both the strands of nucleic acids are anti-parallel to each other. Then with all this information, Watson and Crick, that is James D. Watson and Francis Crick of Cambridge University in England, on the basis of the studies of Pauling, Corey, Chargaff, Franklin, and Wilkinson's X-ray diffraction studies, they proposed a three-dimensional model to explain the structure of DNA. And for this they received a Nobel Prize. The following are some of the important characteristic features of this DNA molecule. The DNA molecule is composed of two polynucleotide strands running anti-parallel to each other, which I had explained earlier. The polynucleotide strand is made up of a backbone of sugar and phosphate. Now this is the backbone, which is formed of sugar and phosphate. The nitrogen base is attached to the carbon 1 position of the sugar. Now this is the carbon 1 position of sugar, this is the carbon 1 position of sugar of the opposite strand. Now this is the 9th position of a purine molecule, and this is the 3rd position of a purine molecule. Always the hydrogen bonds to form a nucleoside, they are attached in this formation only. Each polynucleotide is made up of many nucleotides, hence nucleotide, and these polynucleotides are linked to each other with this phosphodiester bonds here. Nucleotide strands of DNA, they are right-handed coiled helical structure. Now this is a right-handed coiled helical structure because based upon the right-handed or left-handed, we have different types of DNAs. Now this Watson and Crick DNA, we call it as the B DNA. Now B DNA is always right-handedly coiled, and one complete turn of this helix occurs at every 34 angstrom length. Now, one turn means from one minor groove to the next minor groove. We call this as one complete turn. Now, one complete turn, it takes means a nucleotide has a length of 34 angstrom, and in one turn there are 10 nucleotides. So that is why the distance between each nucleotide will be 3.4 angstrom. Now here the measurement is given in form of nanometer. Okay, I'm giving you the terms in form of angstroms. Now the diameter of the helix is 20 angstrom. The diameter of this helix is 20 angstrom in nature. And adenine always pairs with thymine with two hydrogen bonds, and guanine and cytosine always pairs with three hydrogen bonds. The two grooves in the helix, that is the major groove and the minor groove. The major groove has a width of 2.2 mm and the minor groove has a width of 1.2 mm. So these are the specific features of the DNA helical structure proposed by Watson and Crick. Now we shall see about the DNA packing, because DNA is a very long helically coiled structure. Now if we take a mammalian DNA, it has got a length of almost 2.2 meters. Just imagine a 2.2 meters length molecule is just in that small ultrascopic structure called as nucleus. To fit into that small structure, the DNA is highly coiled structure. Now the coiling is different in case of the prokaryotes and eukaryotes. In case of the prokaryotes, because there is no definite nucleus, the negatively charged DNA is held at some places with positively charged proteins to form a coiled loop called nucleoid. And this is the nucleoid structure, and this is the coiled DNA that is held by this protein core. Whereas in eukaryotes, the packing of DNA is much more complex. Now, in eukaryotes, there is, for the packing of DNA, we have the histone proteins and DNA molecule. Now, to form a nucleosome, because nucleosome is a basic unit of this DNA molecule, a nucleosome consists of an octamer. The octamer histone consists of 8 histone proteins, 2 H2A histones, 2 H2B histones, 2 molecules of H3 histones, and 2 molecules of H4 histones. All these 8 histone protein molecules form an octameric histone core, and around this core a DNA double helix of 146 nucleotide pair gets wound up and forms a structure called as a nucleosome. One nucleosome and another nucleosome are held together by this linker DNA, and in between this linker DNA there is a histone 1 protein, and this chain of beads gets wound off into a solenoid-like structure. This solenoid then forms a supersolenoid structure of 300 nanometer diameter, which forms the chromatin fiber. Then this chromatin fiber, during the formation of chromosomes, get further coiled by plectonymic and paranymic coilings to form a chromosome. And the width of each chromatid will be around 700 nanometers, and the whole width of a chromosome will be around 1400 nanometers. So this is how the DNA is packed in eukaryotes. So here I conclude this topic on nucleic acid, structure of DNA, and packing of DNA molecule. Thank you.